Thank you for coming on this little blustery day. I hope I get my flight out later on. All of those stories were true, by the way. And I think probably what I really want to say is, Margaret, thank you, because it really, your first course really did set the stage for just the fascination with plant breeding and all things in that realm. I think the last time I gave a seminar in this room was probably for my dissertation seminar, and that remarkably is about 20 years ago. So I've picked up a couple extra pounds. I've picked up a few more gray hairs, and I've picked up a few good stories about sweet potato. And I want to thank you very much for the introduction. It is a real honor to be here, and I still have very, very fond memories of Cornell. I met my wife here, her major professor sitting in the back of the room, Marvin Pritz. We now have 2 children, and we're both breeders. One does raspberries, and I do both potato and sweet potato. But today I'm going to focus primarily on our sweet potato project, and it's kind of funny that I'm here because I'm almost promoting the stereotype of sweet potatoes for Thanksgiving. But I want to try and break that down a little bit today, and what I'm going to do is kind of take a high-level tour, if you will, of sweet potato and some of the work that we've been engaged with over the years, and it still continues to fascinate me. I really enjoy working. Enjoy working with both potato and sweet potato, but sweet potato, we're really the syndrome of a small fish in a big pond. Sarah said that we're one of the largest programs, but the reality is there's only 2 big breeding programs in the United States, so it's not hard to be the largest program. But I think we also have some global stature too, and I'm proud to say that. And we have a lot of people that come to our doorstep, and it results in a lot of interesting projects. So I'll start now, and I'll wander around a little bit. And I guess I should say, let's see who's in the. I'll say hi to the guys out in Geneva. Good to have you on board. Thank you for coming. So when you think about sweet potato, most often you sort of think about, a nice crop like this. It's a green row crop, and if you get a little bit lucky, you dig up the crop and you get a harvest of nice, good-looking storage root shapes. Now, I was with the graduate students just a little while ago, and you can see where I'm coming from now. They all asked me, what's the best shape for a sweet potato? I said, actually, a brick. And you can see these are pretty blocky, very, very oblong and tubular shaped. And this is probably our model in the United States. It might not necessarily be the global model. But so if you're lucky, you get good yield, and you can harvest those. But I'm here to tell you that actually sweet potato comes in a phenomenal range of shapes, sizes, and colors. And so I really want to challenge you to think outside of the sweet potato box and to see what we can do with sweet potato through conventional breeding and selection. None of this is GMO. This is all conventional breeding and selection. You have the orange flesh types. I think my battery is a little bit low on this. I don't have a pointer here on this. You've got the orange flesh types. You've got the purple flesh up in the right-hand corner. You've got the white flesh types. And then there's this whole other area of sweet potatoes that truly fascinates me. I would have never have guessed we worked in, is ornamental sweet potatoes. And so my goal today, that's a little better. My goal today is just to share with you some of the work that we've done in each one of these realms. I'm going to cover a lot of ground, so I'm going to go pretty quick. So just to give you, I'll talk a little bit about the importance, the global importance of sweet potato, a little bit about the botany and genetics. A lot of people are not familiar with sweet potato as a crop and the genetics of that. And then I'll just touch base on some breeding projects we're engaged with. Sweet potato, of course, is a globally important crop. I think last count it's grown in over 170 different countries around the globe. This is a picture coming from SIP that shows the major production of sweet potato in terms of yield. Of course, China is a major player in sweet potatoes. They probably produce roughly 70 to 80% of the global sweet potato crop. If you step down from that, you come across to Africa. Nigeria and Uganda are either the second or third or second most important producers of sweet potato in sub-Saharan Africa. They're important daily staple crops for millions of people across sub-Saharan Africa. You can see here, these are just dots of sweet potato production and yields. You can see it's widely distributed through sub-Saharan Africa, so it's an important crop across the continent. My prediction is that as we invest more resources into Africa to boost yield in these regions, this equation here is going to change dramatically. Okay, as we boost yield into the green zone, maybe there's going to be more production globally and in Africa. I think that's a real possibility within the next 10 to 20 years. All right. The uses are radically different between China and sub-Saharan Africa. I won't go into those, but just needless to say they are. You jump across here and you can actually barely see it. There's these little yellow dots right here in the US, then certainly in Brazil, in Uruguay and Paraguay, in northern Argentina is major pockets of production. Origin of sweet potato is pretty much around this area here, all right? Central America and northern South America. All right, so tropically adapted crop, but has phenomenal adaptation. When you look at Africa, you can see that sweet potato is expanding faster in area than other major food crops in sub-Saharan Africa. My other crop, potato, is also expanding quite rapidly, but the reality is that when you look at this closely, yes, that's a considerable growth, but yet it only represents about 3.2 million hectares in sweet potato, but it only represents about 1/8 of the area that's under maize in Africa. So we still have a really long, long way to go, all right? In terms of closer to home in the United States, sweet potato is a minor crop. It's a minor specialty crop, but it's growing in importance. And just within the last 5 years, we've picked up several new states that are now producing sweet potatoes around the U.S. Now, is Washington and Oregon a large sweet potato producer? No. But they're trying to produce them. And largely it's due to a couple of new emerging trends, which we'll talk about in a little bit. Sweet potatoes indeed are on the rise, and I think a lot of you in the room probably recognize that. How many of you in the last, let's say, month or so have had sweet potato fries in the restaurants or so? Yeah. I mean, and I talk to audiences all around, particularly in the South. It's almost every hand goes up in the room. So they're really becoming much, much more common in various dining establishments, even at home. Production in North Carolina since 2002 is up about 100%. Up 86% in the US. You can see the crop values considerably up. Even the harvested acres are up. The trends for sweet potato in the US and globally are all pointing in the right direction, and that's up. So you have to ask the question, well, Yencho, why do you work on sweet potatoes in North Carolina? And literally, this is sort of slightly with tongue in cheek. Sweet potatoes in North Carolina really is the Idaho of the potato industry. We're a dominant player in terms of sweet potato production. We produce roughly 45 to about 50%, depends on the year, of the nation's sweet potato crop. So it's a really important crop to our state. We have a very proactive and very well engaged Sweet Potato Commission, and we spend a lot of time there. Stepping down from there, you come to Louisiana, Mississippi, California, and then there's a lot of other minor players that come into play. They're important players, but considerably, North Carolina is really a major player. Sweet potato products are expanding considerably. You're seeing them as frozen products. You're finding them adding color to various drinks. You're seeing them as soups, crinkles, dried product. This actually here is, how many of you have a dog? Have you ever bought any sweet potato chews? I know some guys who are selling loads and loads of kind of sun-dried sweet potato chips as a dog chew. And they're actually quite healthy. And so there's even that. But by far, probably the fun new product that's coming out of sweet potato that's recently come around is this new product right here. It's called Covington Vodka. It's actually named after our variety, Covington, which I'll touch base on a little bit later on. It's a specialty vodka. I think they've really got a great slogan, the best yam vodka on earth. I mean, that does it right there. They came up with a good marketing technique. They've won some awards out in San Francisco, and actually, it's not a bad vodka. I've had it. Pardon?
It's not the best in the world?
Well, you know, I'm not a vodka connoisseur, but for me, if I've had it neat, very, very well chilled, and actually, someone suggested me on a cold winter day like this, you can have it warm. It's actually quite good too, and it's actually got some interesting nutty notes to it. It's probably some interesting nutty backers also. It's actually quite good, but this is made from sweet potato mash, from sweet potatoes that are actually coming from a company called Yamco, which produces a microwave. They use a proprietary technology developed at NC State that uses microwavable technology to sterilize sweet potato and put it in a format of a puree format that can go in a container that is shelf stable. It doesn't need to be refrigerated. And they had so much product left over that what they did is they took some of that mash and they're now making vodka out of it. Where it goes, I know not where, but it's just an example of the new products that they're creating from sweet potato. I don't think this has great nutritional value, by the way. Okay, a lot of my work and where a lot of the growth in sweet potato is coming from is actually from this sector, is the growth in sweet potato fries. It's literally changed my breeding program in the last 5 years. Some for the good, some for the not so good. But the growth in this sector, I have not seen this in other crops, at least as long as I've been around. It's tailed off a little bit in 2013. I can't give you the exact numbers because actually USDA doesn't really track these numbers. So these are some proprietary numbers coming with a group that I work with. It's tailed off a little bit, but the volume is really, really growing. And it's created a lot of buzz, a lot of excitement around sweet potato. But here's the reality. It's impressive. It really is. But it only represents about 2% of the total fry market. So really, it's quite small. But all of the major potato processors are now producing fries, sweet potato chunks, sweet potato mashes, all kinds of new products out of sweet potatoes. And the growth is continuing upwards. And I look at this as an opportunity, not really an impediment at this point. There's a lot of things we can yet to do and a lot of growth we can yet realize in sweet potato. Okay, so that kind of sets the stage for, you know, this crop sweet potato as to what we can do with it, what's driving some of the growth and some of the interest, not only in the US, but internationally also. In terms of botany, sweet potato is it's Ipomoea batatas. It's actually a relative of the morning glory. There's one other species, Ipomoea aquatica, which is of somewhat economic importance, but it's a far cry from cultivated sweet potato. In some cases, primarily in Southeast Asia, and across Africa quite a bit. And I find that increasingly its foliage is used as a food, both for humans and as fodder, largely for pigs, but also for cattle. I see that quite often. So you can use the tops to feed animals and use the bottom to feed humans. It's a pretty diverse species. The batatas, the series of batatas exists as primarily diploids and tetraploid forms. Cultivated sweet potato is the only hexaploid known in the Ipomoea genus. Okay, and its polyploid origin and its exact evolutionary how it evolved is really under fairly significant dispute right now. There's a general agreement that the two species, Ipomoea trifida and triloba, have contributed to the modern batatas that we know, but we don't know this to be fact just yet. You all know this, I think, in the room already, that sweet potatoes, especially in the U.S., are often called yams. This is incorrect. True yam is a Dioscorea species, and they're actually monocots, so it's not correct to call them yam. I won't go into why that persists in the market, but if you look closely on, say, if you see a can of candied yams that you're going to pick up for the Thanksgiving holiday here, I'd say first, don't pick it up. pick up regular sweet potatoes and do something with them other than the candied yams, but you'll see somewhere on that label sweet potato products, all right? In terms of the genetics, it's a fairly complex polyploid. Base number of chromosomes, 15 chromosomes, and it's a hexaploid, which makes breeding both very, very frustrating, but also very, very interesting at the same time. The chromosomes are very small. There hasn't, as a result of which there hasn't been a lot of cytology work done. There has been over the years, but it's hard to find a good cytologist these days. Highly heterozygous, complex cross incompatibilities, which makes selfing batatas very difficult, sweet potatoes very difficult, and very often I can't make the cross that I want to make because of incompatibility. So we have to do a lot of work as sweet potato breeders to get around that. A lot of controversy in terms of whether it's an allo versus an autopolyploid. I personally believe that it's an autopolyploid. I think we've shown that with molecular markers, and I think that's probably the conventional wisdom. Compared to my other favorite crop, potato, there's a lot of wild species out there, but largely they're relatively inaccessible. So going to the related germplasm of sweet potato as a source of useful genes generally has been very, very difficult and has not really been exploited, certainly like it has been in potato, okay? So that's been a big issue for us. In terms of molecular genetic resources, I'm not going to go into all this list right here, but suffice to say that the genome size is somewhat large, but not super big. There hasn't been a lot of molecular genetic work done in sweet potato. If you compare it to, say, rice, maize, potato, the grains, we have a long way to go. And I'll touch base on that here towards the end of my talk. With the McKnight Foundation, we've done some great work. I was hoping that Rebecca would be in the audience today. I know she's traveling, as she and I are very similar in that respect. We have had long-term support with the McKnight Foundation now coming up on almost 12 years. And it has been a good project, largely based in Uganda. And our focus has been with the National Agricultural Research Organization, the National Agricultural Crops Research Institute, NACRI, NCSU, and University of the Natural Resources Institute at University of Greenwich. And our goal there has been to develop high dry matter orange flesh sweet potatoes to combat vitamin A deficiency in sub-Saharan Africa. We also are concerned with these pests right here. Two pests of great import are actually sweet potato virus disease, which exists as a complex of two viruses, sweet potato feathery mottle virus and sweet potato chlorotic stunt virus. Together they co-infect the plant. This is a healthy plant right here, and I've seen outright death of the plant. We do not have that complex here in the United States, so we don't wrestle with that issue here. The other issue that we have, which is of great consequence, will devastate an entire crop, is sweet potato weevil. Trying to develop resistance to sweet potato weevil has been very, very problematic, but the project is making some good progress on that, and I'll touch base on that a little bit. North Carolina's contribution to this project has been developing molecular genetic resources, first generation molecular genetic resources for sweet potato. I've had two students on this. Jim Cervantes actually is now a breeder with Vilmarin. He's a carrot breeder, and I'm just about to finish up a second student right here. Bernard Yada is just going to be coming back to the U.S. to write his dissertation here within the next couple of weeks, actually. Goretti Semakula is the project leader in Uganda. Robert Mwanga was actually my first student and is now the lead sweet potato breeder for East Africa. This is just to show you that we have successfully constructed a complete genetic map of sweet potato using AFLP markers. It's the technology that we had at the time, and I think it's no small feat this is largely Jim Cervantes' work, that he was actually able to develop a linkage map for all 90 chromosomes. That was a heck of a job. This is the map for Beauregard, one half of the parent. He had to do it for the other parent, Tanzania, so we have two maps like this. Using both duplex and triplex markers, we've been able to align what we believe to be the homologs of the various linkage groups. Then we developed a system to name them and to begin to sort through those. This would just be what we consider to be linkage group 1 and the 6 homologs. And here are the various colors dictate whether it's a duplex marker or a triplex marker. And we can use that by inference to align the homologs. And this is what really led us to believe that it's probably an auto polyploid, and there's segregation amongst all 6 groups. We've used it. We've used it to tag QTLs. This was the first in sweet potato. This actually is a QTL. You can see it's a big Herking QTL, and it's not highly. It doesn't explain. I think it's maybe 15–20% of the variation in the trait, but it actually made me feel confident that what we were doing, because as a breeder, I've always known this is a QTL for dry matter, this is a QTL for starch. And I forget exactly how, yeah, this is, if there's a line under it, this QTL resulted in lower dry matter content and lower starch content. As a breeder, I've always known it's difficult to breed high dry matter, high beta carotene lines. And we have shown that molecular genetically, because this QTL for beta carotene right here actually results in increased beta carotene. So we have that negative linkage. And that's something that I think we've broken now in the sweet potato breeding programs in East Africa. But, you know, are we using this as a tool? No. And do I expect that right now with the AFLP map that we have in place that we can begin to reasonably use it to facilitate marker-assisted breeding in sweet potato? No. I think we have a long way to go. And I'll touch base on where we want to go in the future with that. But this was the first sort of demonstration that, yes, we can do the same thing that many of the other crops have been doing altogether if given sufficient resources. Okay? So I feel good that we could construct a linkage map and that we can tag QTLs and we can demonstrate this. But I'll be honest with you, I feel much better about this picture right here. And this is actually a picture of a grower, a farmer in Uganda, who actually is growing orange flesh sweet potatoes. And this was his house. And through growing orange flesh sweet potatoes and selling the crop and actually selling seed, he's now been able to do this: build himself a new house. And he's sending his kids to school. Now that, to me, is a success story. So I kind of rest my hat, and really this isn't our work, it's really the work of our collaborators in Uganda, showing that orange flesh sweet potatoes can contribute to nutrition, can contribute to livelihoods in sub-Saharan Africa. And this is just one picture that I'm showing, but there could be many, many more examples of that. So, you know, I like this, but I'm really proud of that. All right? Okay, so let's talk a little bit about the breeding work that we've done with sweet potato. And largely this is just going to be a smattering of various projects that we've been engaged in. But I think probably as breeders you'd be kind of interested in what does a breeding program look at NC State and how do we do it? And this is my attempt to kind of boil that down into one sort of flow diagram for you. We start up here with recombinations, and we use a combination of both polycross and paired breeding nurseries. And we generally start out like a potato breeding program, like any clonally propagated breeding program, with a single plant. And in my group, we try and plant about 60,000 unique individuals in the field every year. Those go in spaced usually about maybe a ½ meter or so apart. We'll start out with about 60,000. We'll whittle that population down in year 1, select about 2 to 4% of those. We'll whittle that population down to about 1,200. Do I wish I could select maybe more like 10% or 20% and I'd feel better about it? Yeah, maybe so. But this is the way that it's working out right now for us. In year 2, this is where things differ quite a bit. We take those roots that we harvest, those are roughly 1,200 to 1,600 roots that we harvest in year 1. We'll store those, and then in year 2 we'll do a process called bedding out, where we plant the roots in our system. We use a small tray, and we sprout off of those roots. And that gives us lots of plantlets that are clonal, right, that we can use for planting in the second year. In the second year, we plant out 2 25 plant plots at 2 different research stations in North Carolina. They're separated, they're in two rather distinct environments and separated by about a 2-hour drive or so. We'll take this 1,200 take about 10 to 15% and whittle that population down to about 120. And in cycle 3, we now have sufficient quantities of roots that we can plant out anywhere between 3 to 500 to 150 plant plots, which we put out at 3 to 5 different locations around the state. And we exercise selection at each one of these locations, irrespective of what we pick up at those other sites. So we pick everything at each site, and then we compare notes, which ones were selections that were selected at sites 1, 2, and 3, or so on. Get an idea in terms of stability. By about years 4 to 6, we're ready to invest in the clones a little bit more deeply, looking at replicated trials, and that could be in at least 5 to 8 sites. And at this point, we're ready to probably place them into tissue culture for virus cleaning. Because remember now, every year here we're picking up pretty much viruses, which are impacting yield and quality also. And so at about this stage, they'll go into tissue culture, and then it'll take us maybe about a year to clean them up into tissue culture. They come back out, and we start to go through the variety. We'll start to trial them during this period of time quite a bit. Hopefully we'll come out with 1 or 2 new varieties that we'll release to farmers. This MPUR is our micropropagation research unit, and the MPU is along with the North Carolina Crop Improvement Association and the Foundation Seed Producers, tasked with providing propagules of true to type, high quality planting material for our certified seed growers. So we have an industry in North Carolina that's focused on producing high quality seed of sweet potato, and we have a limited generation program in place. Probably our seed system in North Carolina, I think, is one of the most sophisticated around the globe, to the best of my knowledge. We do it well, and a lot of people come to us for seed all across the states. There are 2 steps that I really enjoy. This step here and this step here. Part of our program is actually conducted on farm through what we call the Grower Participatory Breeding Project. Very similar to what a lot of my colleagues do in Africa, we do it on farm with growers in their fields. And we come in and we'll plant both single plants, our first cycle of selection. I'll go back, and we also produce this generation, early generation materials in farmers' fields. The farmers help us plant it. They manage the fields according to their methodologies. We come in at the end of the season, we mow the vines, and we dig it in collaboration with our partners and in extension, and we make our selections on farm, and we evaluate the materials with the growers. They give us feedback. Generally, we're working with seed growers. Seed growers like something, they see something they really like, they'll pick it up, they'll keep it, and they'll start to move it forward. And we'll have an MTA process in these days to kind of paper trail all of that, because now all of our varieties are plant patented when they're released. So here's the grower right here. These are 3 members of my team right here, and this is one of our extension agents that's in the field with us. And it's this project now, I think, is going into its 15th year, and it's funded through the North Carolina Sweet Potato Commission every year. And our growers have really bought into it, and it's a good way for the breeding program to stay connected with the industry and to understand what their needs are and what issues they're facing as an industry. Sweet potato flour, remember it's a morning glory, so if any of you have seen morning glories, this is a picture of a typical flower. It's hard to make crosses with. Because it's very difficult to cross, many breeding programs have resorted to what we call polycross breeding nurseries. John, where are you? This is what I told you need to see. Yeah. So this is what one of our polycross breeding nurseries looks like. Basically, we select superior genotypes, and we plant them out. This is one block of about a 6-block planting. And we select out superior genotypes. We allow mostly bumblebees and honeybees do the pollination for us, and we work with half-sib families. Okay? Basically, every cross that I make in sweet potato can result in a maximum of 4 seeds. And functionally speaking, usually we only get maybe 2 seeds. And maybe about 30% to maybe 50%, maybe 60% of our crosses actually take. And so if you want numbers, you've really got to either have a big team that's making a lot of paired crosses for you, or you resort to something like this. It works for us. The Ugandan program and most every other sub-Saharan African program is doing the same thing, but they have more hands, so they're doing more paired crosses. This is what our nurseries look like in September. You can see some flowers. We trellis them up onto hog wire because when I first arrived, they used to run them down on the ground, and I decided I'm not going to be bending down all the time. We actually trellis our plants, and we can pick the seed, get much better quality seed that way. This is what a fruit of sweet potato looks like, this little green capsule right in here. You'll get a maximum of 4 seeds in there if you're lucky. This is Ken Pakoda, one of my associates, picking flowers off that trellis. You can see Ken is 6 foot tall, okay? So it's actually a pretty big group, and it takes a lot of time and effort to sort of weave your plants through this trellis. And we also use a root induction Ipomoea setosa plant to induce flowering. So we graft most of our Batatas materials onto a flower-inducing rootstock. For us, it's called Ipomoea setosa, Brazilian morning glory, and this induces flowering in our rather longer day environment. Various breeding programs around the globe use various species. For us, setosa works quite well. Again, it's a morning glory. So the flowers open in the morning, they close in the early afternoon. There's genetic variability for this, but usually your best crosses are going to be made in the morning. If we're making crosses in the greenhouse, which we do, we're making crosses usually before 10 o'clock is when our best success is. And after 10 o'clock, we have some issues. If we get lucky, the seed will set fruit. This is green fruit right here. We come in and we pick the seed right here. These are ready for picking. Once that little minor pedicel right there starts to brown and darken, you want to get in there, you want to pick the seed. Very often different genotypes will shatter, and so they'll spit their seed and you'll lose that. So you want to get in there before this opens here, and then we just pick those and put them into a bag. And we're keeping records of the mom. We don't know the dad. Not yet, anyhow, in this format. I have found that we do a thing called frost rescue. When we see the first frost coming on, what we can do is, I can back up here, oh, it's hard to see. This peduncle right here, you can see them right in here. We can come in and we'll just harvest lots and lots and lots of fruit that hasn't quite matured yet, and we'll bring it into the lab. This actually happens to be at my house. I had a nursery in my backyard. And we can finish the seed off in the house under those conditions. About 1/3 of our seed harvest every year comes from this technique. And there's a lot of breeding programs don't realize that you can do that. And we've really, a lot of folks come and see us and we say, oh, you can do this. And they're just like, oh my gosh. And so it's really been a big benefit for us. It's just one of those little tricks of the trade that I think it was Mark and I were talking about before. You don't learn in the books. You learn by doing. We've had some success. We've had some success. We released a variety early on, and this might be my one great success here, Walter, I don't know. In 2005, pretty early on in my career, about 8 years into it or so. It's called the Covington sweet potato. This is the trajectory of Covington. Actually, we selected it in my second year as a single plant in 1998. It actually is a graduate of our grower participatory breeding project. So it actually came from on farm. And fast forward to 2011. Last year, I think we had close to 52,000 acres of Covington planted in just North Carolina alone. It represents probably about, which is scary, probably about 85 to 90% of North Carolina's acreage. I would really rather growers plant a more diverse set of varieties, but this is the tradition in the sweet potato industry, dominance of one variety until they find a new one. A 350-fold increase in 8 years. As a potato breeder, I am terribly envious. And it's all because of that vegetative propagation cycle through plant vegetative cuttings, not through the storage root increase. You do that with sweet potato also, but you can also do it via plants. It represents roughly about $160 million farm gate value to our economy. That's just the farm gate value. That's not the added value with the other things. So it's made a big impact, and our growers have been very, very appreciative, and it's gotten us a lot of notoriety. We patented in 2005. It's the very first commercially successful sweet potato variety that's been patented in the U.S. And that process, walking that line through that process and establishing a licensing arrangement and getting buy-in from our growers and the whole industry contributed significantly to this. But it's done, and my message to the growers back then was, if we don't do this, we will not be around in 10 years' time. And my phrase to them was, I want a breeding program that can thrive, not just survive. And I feel strongly that had we not done that, we would not be around. And I think the industry now recognizes that. They see the value in it. So we've worked through that. This is the prior dominant variety called Beauregard. It still persists. It's still quite popular in the Deep South. You can see the shape. This is really very representative of what we would typically see in Beauregard, North Carolina. This is Covington. And I think you can see immediately, right off the bat, higher yield, more consistent shapes, and that resulted in about a 30% increase in pack out. And that means dollars in farmers' pockets. As a breeder, I also need to realize that you can have great disappointment. Hatteras came shortly after, 2 years after. Beautiful sweet potato. I still cry when I walk the plots when I see Hatteras. Very drought tolerant. Great shape, like Covington. We released it. It quickly ramped up. I think we had somewhere around maybe 5,000 acres. It came down with susceptibility to a syndrome called internal necrosis in storage. Completely, every grower walked away from it. It just had that one minor flaw, actually a major flaw. Yeah. But one key flaw. We use it as parent, we use it as a check variety very often, and it performs beautifully, but it just does not store. And we had released it. I think there's a colleague of mine said, "Craig, great name. What a waste of a great name." Yeah, you know, I mean, if you've ever been to the Outer Banks, Sarah, I know you have. It's a wonderful place. So we named it after a spot in the Outer Banks. It's very dear to my heart. It's gone. Hatteras persists, but not in the sweet potato form. So that, you know, we had a great success here. We thought we were on to success number 2. Bang, we got shot in the foot, and now we're still trying to replace Covington, but we set the bar quite high. Okay, so this slide here is just to kind of show you, these are all sweet potatoes. They're all sweet potatoes. They come in a wide range of shapes and colors, and it continues to fascinate me. I was talking to graduate students a little bit before. They were asking me about this thing, shape, color, can you breed for it? And I said, yeah, you can. If you want me to breed a round baseball-like sweet potato, I believe we can do that. The chipping industry might be interested in that. Just give me some time and some money, of course. I think we can do that. This here is actually the predominant interest right now, but there's a lot of interest in sweet potato. And here's the thing that just really blows me away as a breeder, and Margaret, this is what captured my imagination, is variation and segregation. And here's the thing that is really cool. So this is a cross. We did an NC2 mating design several years ago, and we crossed an orange-fleshed sweet potato with a purple-fleshed sweet potato, okay? And this represents, what we did is we decided that we were going to take pictures. This is, we looked at 20 progeny in, I think it was 25 families. This is the cross between L96 117 and 415. L96 117 is about like this. NC 415, this is purple here, this isn't black. This is a purple clone. So these are the F1 progeny from that cross. Go figure. Huh? Look at the segregation in there. Now, and you don't even get sort of intermediates. I mean, you just get this phenomenal type of segregation in sweet potato. And so what fascinates me is, well, think about the opportunities in a cross like this. You know, in this day and age, in terms of functional foods, you've got anthocyanins, you've got beta-carotenes, you've got high phenols, you've got low glycemic index, you've got new flavors, you've got exciting colors, different starch, sugar profiles. low acrylamide levels. This is something that really the French fry industry right now is very, very all kind of concerned about. All of this exists within this material. The thing is the challenge is how do you decide what to develop, and how do you get there? Each one of these projects is a 4 to 6 year project, maybe more. So how do you as a breeder decide what you want to do? And how do you develop a vision to develop this new crop? Maybe some people haven't even really thought about it yet. Well, the sweet potato processing industry came to us about 5 years ago, and this has really changed the focus of our program. We're now working to try and determine the important traits that affect processing quality. We know this quite well in potatoes, so we have an opportunity to learn from potato. We know it from some of the other major processing crops, but we still have a lot to learn in sweet potato. These are the primary traits that appear to make a sweet potato French fry very important. They're all also important in terms of table stock industries. So what has actually happened is my life's got a lot more exciting, but it's also gotten a lot more frustrating too, because my job has not gotten any easier in the last 5 years with the emergence of this processing sector. If anything, it's gotten much, much more difficult because now I have more traits that I need to track and understand. Ken and I recently tallied this list up a year or so ago. When we started back in 1997, and we were looking at about 28 traits that we thought that we'd routinely evaluate. And I think we're up to around 43, and actually this list is a little bit shorter because there are some traits here that are composite traits. But we're actually evaluating more traits than we ever were with less funding and less resources. How do you do that? I think partly it's adopting what many of us now call as an entrepreneurial spirit and teaming up with various people to help you do what you need to do to be successful. Part of it's just brute force. Using my experience as a potato breeder, we now screen a lot of our materials with our collaborator in food science for friability. And this is just a bunch of sweet potato chips of various clones. Here's our Chex. This is how Covington does. Evangeline, another somewhat popular clone, and Beauregard. You can see they're very dark. The chip industry actually wants a nice bright yellow chip. This would example of something like that. And these are just second year materials that we're screening en masse for their color. This gives you an idea of we started this project back in 2006, and you can see that we've really begun to ramp up our screening in response to a demand from the industry. And we're moving the curve the way we want it to go. So the yellow bars are where we started in terms of frequency distribution, in terms of what this rank down here is the Snack Food Association rank. It's actually kind of a measure of the browning or the brightness of a chip. We want bright orange chips, and we want them to be down around 1 and 1.5s. And if you can see, when we started in our breeding populations back in 2008, we were kind of skewed over here into about the 3.5 range, and we're making progress. As of 2011, we have a good pool of materials now that we're now beginning to intermate and developing brighter chips. So we're making progress, and I think we're making progress fairly quick using kind of old school tools. But the big issue for the chip industry is how well does a fry color relate to how well does a chip color relate to a fry color? Well, the simple answer is not very good, but it's the best we have right now. So we use the tool, we use the lever that we have to do the job that's at hand right now. But we've brought some new tools to bear to this problem. We're trying to develop an understanding. We know that sugars are related to browning. And so now we've brought a new tool to our program, which we use quite effectively, called NIRS, Near Infrared Reflectance Spectroscopy. And what we've done is we're now using NIRS to quantify the sugar profiles, the starch and the dry matter profiles, beta anthocyanin profiles in almost all of our sweet potato lines. And we're scanning using NIRS now about 2,500 clones a year. And then we use that information to help us determine which clones we want to advance, which clones we want to fast track, and which clones we might want to intermate. This is just a printout of an outputted format from NIRS. These would be the second year clones here, and we're scanning for amylose, dry matter, fructose, glucose, moisture content, starch, sucrose, total sugars, and we can bolt on beta carotene. You can bolt on iron and zinc too. The International Potato Center has curves for each one of those traits. This is in one scan of a freeze-dried sample, and it really works well for us. It's done on a freeze-dried sample. Our next goal is to try and do it on raw samples. That's a little bit hard to do because of the latex in sweet potato, but I think that it might be done. Ideally, we would do that in the field, but I don't have that technology at hand. I'm guessing Monsanto does, but I don't as a sweet potato breeder. But this tool for us, it took about 2 years to develop, and in the last year and a half now, we're using it routinely, and it's really benefited our breeding program. And we're bolting on some other things on the potato side to do the similar things with potato. And I'm optimistic that we'll get that job done. All right, so that's the processing. Let's change gears real quick and look at another project that I really have enjoyed, and that's developing purple-fleshed high anthocyanin in sweet potatoes. And this is just sort of a bar graph of what you can expect in terms of milligrams anthocyanins per 100 grams fresh weight. You can see blueberry really produces phenomenal amounts of anthocyanins. Red cabbage, a good source of color. Blackberry is a good source of color. I don't have up there red beets, should be in here. Would be pretty high, I would imagine. And you can see here that actually purple sweet potatoes do pretty good too. But the thing that purple sweet potatoes do that all of these can't do is produce tonnage. Lots and lots of color per acre. And so we've teamed up with a company who's interested in using anthocyanins derived from sweet potatoes. The anthocyanins from sweet potatoes have some unique stability characteristics for the food processing sector. And so we've teamed up with a company who has a proprietary technology to get out the color of sweet potatoes. And then they use that and they concentrate those anthocyanins to what they call an EV30 standard, which is an industry standard, and then they sell that to various entities in the natural colorant sector. And the whole drive here is to try and get away from the synthetic FD&C reds and blues, which have some issues with children in terms of hypertensive activity disorder, etc. Also, our major sources of natural colorants are coming from overseas. Some of those sources are somewhat suspect, and there's worries about adulteration of those sources, and so there's a drive to sort of inshore color production in the US, and we're trying to help facilitate that. These are a bunch of lines that are segregating for purple flesh. We literally went around the globe searching for germplasm that produced anthocyanins. Much of it was unadapted, very poorly adapted in fact, and we intermated those with a lot of our materials, some of our white fleshed lines, some of our purple lines, some of our orange fleshed lines, and we are now just recently released last year our first variety of colored line. This is purple 06020, and this is licensed exclusively to a company called Avoca in North Carolina, and their whole goal is to extract the color out of this material. We know a fair amount about the anthocyanin profiles that are coming out of sweet potato. Right now, our next goal is to change those profiles in response to the needs of the natural color industry. It's mostly pionidin and cyanidin with a little bit of pelargonidin in there, but we see in our segregating progeny that these ratios change, and those ratios result in different hues and colors, and we can modify those using conventional breeding. Markers would help us a lot more, but so far we're using biochemical assisted breeding to do this. We have NIRS scans for this on total anthocyanins, but our NIRS scans can't give us individual anthocyanins. This is part of the team. This is Dr. Den Trong. He's a USDA ARS scientist based at NC State in the food science department. Den is a great guy, and we do a lot of work with him. This is one of my other co-breeders, Ken Pacoda, who's a research associate, researcher in the department, and Ken is one of the best breeders that I know. Run circles around me. And so we've done a lot of work together. Ken and I have been together now for 15 years. We have a seed system in place for 06, 020. This is our first seed harvest on farm with a seed producer. And I believe that we're on track now to ramp up to somewhere north of 2,500 to 5,000 acres next year. And then a good portion of that will actually get crushed. And then we're going to have our first large scale sales of this new product in North Carolina. All right, I think we're in a good track here. So the last thing I want to talk to you about is actually, a project I would have never ever guessed that we've been engaged in, and it continues to surprise me till today, is this thing called ornamental sweet potatoes. And I think probably if you're reasonably knowledgeable and you travel around, you keep your eyes open, you have seen ornamental sweet potatoes here in Ithaca. I know I have. And you've seen them all around the country. Are they right up back? Yeah. I'm proud to say that there's a good chance that some of those might be ours. There's 5 new companies, big companies, that jumped in on it after we started. But I want to tell you the story about that because I think it's kind of a cool story. So how does somebody get from this, where your sole focus is on producing new storage routes, to this? How do we make that leap? Well, it's a funny story. And it actually begins with this guy here again, Ken Pacoda, who's remarkably creative. And it's just hard to hold Ken back. And this is our first wave of varieties that were released. These are the Sweet Caroline series. But actually where Ken started at with this project is right here in the field. And what Ken actually wanted when he started to make these original crosses is he wanted a spacer, a visual marker between plots that he could work with. Because what happens in sweet potato and many of your crops, once the crop grows, you can't really see it. I don't really see great segregation up here that I can walk through and immediately say, oh, this plot stops, this one ends, visually from there. And Ken wanted to do that, and he came up with the idea of going for a colored sweet potato. They were out there, and he thought, well, we can just kind of put in like maybe 3 plants at the end of the plot, and we'll visually be able to see that, and that'll work out really well. And I was like, well, Ken, I think that's kind of a little bit impractical in terms of planting, because we plant on a machine, but I said, go for it, And so we tried that, and it really didn't work very well at all. But Ken is very curious. And he said, well, what happens if I start making crosses, just out of curiosity, this is the sulfur and the blackie. They were out floating around the industry for years. We don't really know where they come from. We think we think Southeast Asia, but we're not really sure. And he just started just out of curiosity, making crosses between the cultivated types, our table stock germplasm at that time, and the various types that we had on hand. And I could still recall Ken coming into the office and saying, hey, Craig, I want to try this. And I'm sitting thinking, we don't have a lot of resources. The Sweet Potato Commission wants us to develop a brand new variety like yesterday. And you want to monkey around with and I just couldn't stop him. I said, give it a try. I'd like to see what happens. And he kind of went in and went off, and he would be in the greenhouse on Saturday mornings and whatever, and just making crosses. And we're in a horticultural science department. And what was really kind of neat was the floriculturalists and the ornamentalists in the program. We kind of talked, and we'd kind of share notes occasionally, visit in the greenhouse. They started saying, wow, Craig, these are actually pretty pretty. Have you thought about doing something with them? And I said, well, yeah, kind of interested in seeing if there's an opportunity there. They helped us invite a series of ornamental people coming in from some major companies. We had about 5 companies come in, and we just kind of shopped the materials around. And they got excited. And what happened was the ornamental companies came in, and they gave us the vision. We had no idea about how to market something like this. We could see that it was kind of cute and pretty, but we had companies come in and they said, oh, we can take this color series, market it as a series, and we can use this as an outlet. And the first group that we teamed up with was a group called Bodger Botanicals. And this is at their PAC trials in California. And they really jumped on the bandwagon. They said, look, we'll help support your activities. In exchange for that support, we want to have the first option to market your ornamentals. We developed a kind of a license arrangement with them. It's paying for a couple technicians in the program, and we get royalties on the backside. And this is one of the displays they made early on in the ontogeny, if you will, of ornamental sweet potatoes. And they really hit the ground running. We were the first wave of ornamental sweet potatoes called Sweet Caroline series. And now we have this part of the breeding program that occupies a space of about 4 to 6 acres every year that is now ornamental breeding. And we now have an exclusive license with a company called Proven Winners. They're one of the best known brands in the gardening industry who helps to support this activities. And what I really find interesting in here is this is actually a backdoor way to capture value in the ornamental sector, which might have value in my table stock sector. Here, I can get interesting disease resistance traits. They can be in this area, and I can use that, and I can cross these into my Batatas materials. And I don't have a lot of time, but I can tell you a good story about that shows that. But now we have the Sweet Caroline series, which are a series of unique colors, deeply lobed leaves right here, the Sweet Caroline purple, Sweet Caroline red, Sweet Caroline light green. We have the Sweet Caroline variegated series. We have heritable variegation that we have segregating our progenies that we can use. We have the Sweet Caroline Sweetheart series, the same color palette, if you will. I actually like these quite a bit in the purple, the red, and the chartreuse or lime green. We have the Illusion, Sweet Caroline Illusion brand, which has the very, very deeply lobed sweet potato varieties in the same color series again. And then we have what we call our onesies, Sweet Caroline Green Yellow, which is variegated. We have both female inherited, that would be mitochondrial inherited variegation. We have other variegations, it's just we don't know if it's transposon or what, but we know we can breed for it. And then we have random mutations in our populations. Almost sounds like maize a little bit, doesn't it, Margaret? Yeah. Which is our variegated species. We have these, what we call Sweet Caroline Bewitched, is these toothed genotypes which have a unique appearance that are marketed. And this is just to give you an idea what we see in terms of the color and the lobing. And uses, you know, this is a couple of different uses. This is actually out in Chicago. You know, you can put them in potted plants in mixed plantings. Hanging baskets, quite attractive. We make some of these and we give them to our chancellor for the chancellor's residence every year. We really make a hit with the chancellor. We get some good invites to some soiree parties that way. You know, you got to do what works sometimes. But he really appreciates. He's a horticulturalist, so that works well for us. You can have these. A lot of stuff often is cascading out of pots, which is actually quite attractive. And really, the two drivers of this right now, I would consider myself not. I've been more of the business entrepreneurial side of it. I've been the facilitator of this. The drivers of the innovation and the variation is really Mary and Ken. And really, Mary Reber these days has a BS degree in horticulture, but boy, is she passionate about ornamental sweet potatoes, and really loves to make crosses and knows what she's doing. Just learned it through the seat of her pants, and now really. Works very diligently in this area, and is a key go-to person as a liaison between us and PW. PW, Proven Winners reps, come to our fields several times a year, and we go to their fields. And what it's resulted in, we have some new varieties coming out, but to boil it down is, I would have never have guessed this. We have a footprint of 15 varieties in 11 countries, and Proven Winners is our exclusive distributors of those. I think we sold north of 5 million plants last year. And I would have never, ever, ever have guessed that we would have been doing this with this crop we call sweet potato. It's a it's a testimony to the crop but to the variation that exists in the species. And it's been really fun. So where do we go from here, I guess one says. And I think the next horizon for us is going to be, and I think we'll probably be doing it with some collaborators here in the room and probably some new ones that we picked up here over the last couple of days. I think we can do a lot better with sweet potato. And I think it's time for us to invest deeply in molecular genetic tools to facilitate sweet potato, largely to feed people right now. But these tools can be used in a lot of different venues. I think that we all know this is the results of a sweet potato convening, a sweet potato genomics convening that several of us participated in out at the Gates Foundation earlier in the year. The Gates Foundation has invested in a program called the SASHA Project, which I've been very fortunate to have been involved in, Sweet Potato Action for Security and Health in Africa, which has basically built a whole breeding platform for sweet potato across sub-Saharan Africa. They've done a great job. It's been led by the International Potato Center, and it's called SASHA, and I say SASHA 1 has been very successful, and these are some of the results that are coming out of SASHA 1. This is the lead team. It's led by Dr. Jan Lowe. I think Jan's actually a Cornell grad too, if I recall. Cornell undergrad, right?
I know that she's a grad, I'm not sure.
Yeah. Jan is a force of nature to be reckoned with. Here are some of the breeders over here: Wolfgang Grunberg, Maria Andrade, Robert Mwanga, NC State grad right here, virologist, and just this is the launch right here. And the SASHA project is to be great strides in these 5 key areas. The transgenics hasn't gone so well, so that we can sort of downplay that a little bit. But what we did is we came together in a convening, and we just pitched the project to the foundation that's focused on developing a new set of molecular genetic tools for sweet potato. And I do this sort of my eyes wide open, sort of realizing that sweet potato is very, very complex genetically speaking. But I very firmly believe that it's important for us to begin to build the core genomic infrastructure for sweet potato and bring other smart people to the table to begin to work in this fascinating crop. The program right now is we will sequence a diploid sweet potato that is actually developed in my laboratory with Dr. Zhangjun Fei at BTI Cornell. We've started to do that work already, developed some bioinformatics platforms with that. We have NCSU and the University of Queensland involved in that. We want to look at genotyping by sequencing in sweet potato. We've had some good conversations with the cassava team around that topic yesterday. Some databases and bioinformatics that will go for that, and then of course the breeding and capacity development in sub-Saharan Africa. I think that we can probably keep the foundation engaged and supportive of us. For me, there's 2 major components to this project. It's one, human resources. We need to develop the capacity, the people to do the work. We have a long way to go in sweet potato in that realm. And then we need to develop the genomic resources. And I won't go into the details right now just because I think I'm a little bit out of time here. But we're doing some good work so far. So far, this is initial data that's coming on our diploid, the KMER distribution. I think we've identified a suitable line that can serve as the reference genome for sweet potato. It's relatively clean, so it's not contaminated with all kinds of bacteria and viruses that'll mess up the sequencing work. And so we've begun that work, and we'll see where that takes us. I'm not going to go into detail on this, but I think every one of you in the room know that doing work like this takes people, a lot of really good, smart people and dedicated people. And this is just a partial list of all the groups that I've been very fortunate to work with over the years. And I think with that, I hope that I have now given you a new appreciation for sweet potato as you go forth and you eat your Thanksgiving meal. Think just a little bit more deeply about that. And I wish you, I'd be happy to host questions. I've gone a little bit long, but I like to tell the sweet potato story. I'd be happy to entertain any questions that I can, but I also want to wish you Happy Thanksgiving, and I want to thank you for the opportunity of coming back to this great institution. It's a pleasure.